Welcome back to this channel. In this tutorial, we'll be looking at multiple online tools and web apps which will be useful for biologists. So most of them will be AI based tools and some of them will have machine learning algorithms, so which is not completely AI based tools, but you can use them for a number of research and analysis works. I am a computational biology and I work on the cancer and related data set, and I use a couple of tools. Of course, I don't use all of them. Here I have listed 29 different types of online AI based tools, and but I use some of them like string, then enricher, all these certain tools I use, but I will like to present here more than 20 exactly 29 different online tools which basically involves the literature review and analysis and multiple other AI based chat engines and all other than chat GPT and Gemini. So there are certain other tools for protein and structural determination, then determination of the drug structure or the chemical structure. So we'll discuss about these different tools in these tutorials. So to give an overview, uh, I would like to uh, list down what are the different types of tools we'll be using here. So we'll be using for literature review, like one of the most like very less known tool is Elicit. So we'll like to explore that. Then we'll go PubDater, and which basically used for extracting the genes, disease, proteins. We have Cite, which validates the important papers. Then Ensemble will be used for downloading the gene protein sequences. We have Alpha Missense, which predicts the pathogenic mutations. Deep Variant, which calls for the variants from the sequence data. And we have Splice AI, which assesses effects on RNA splicing. There is DeepLock2, which predicts the protein localization. We have AlphaFold or Robeta, which predicts the 3D protein structures. Protein MPNN, it's used for designing and optimizing the protein sequences. We have String, which basically is used for the interaction analysis of protein-protein interaction network. We have another database called MetaboAnalyst, which basically analyzes the metabolomics data. MOFA to integrate the multi-omics datasets. And XPC, we have Biopsych and SIB, Informatics for Swiss Institute of Bioinformatics, which is used for the functional and the pathway annotations. We have DeepCell and PathAI for analyzing the medical images or microscopic or pathology images. CRISPRNet to design the CRISPR guide RNAs. We have DeepChem or Atomwise for virtual screening and AI-driven drug discovery. ESM for Metagenomic Atlas. ESM Metagenomic Atlas for exploring the predicted protein structures. NeuroSnap for specialized AI analysis and visualization. And we have Eterna or Prompt Bio, which use in RNA design and AI-assisted biological workflows. So to begin with, we'll first introduce the different tools one by one, and maybe we'll also do a small project to identify the disease associated with the gene variants, their functional impacts and the protein structure, and also interpret the interaction network and the drug discovery using the AI tools. So maybe this video will be too long, so in this video we'll be first understanding the different tools which are there. So some of them will be also GitHub tools which we might have to download it and customize it according to our needs. So others will, most of them will be online tools, while some will be some offline uh, uh, like tools or softwares. So we will like to try out all these tools in a small project, which we'll maybe discuss in the part 2. But in this part we'll discuss about the different website, and we'll see what are the features these tools basically offer. So the first one for literacy survey is Elicit. As you can see, this is the interface for the Elicit, and the website is https elicit.com. So it's used for AI based literature review. Basically, it summarizes the abstract and overall work which is done in the paper. So basically, chat gpts and Gemini, they are uh, one of the tools similar to this Elicit, but it will basically give more concrete results and more and it will narrow down your search and optimize your requirements. So this basically has different purpose, like scientific literature and research papers summarization and answering like answering your research questions or hypothesis. So how to use it? Basically, it's a free tool. So we'll try one example, and there are certain advantages. So basically, you can try out different papers and different papers discussing about our requirements. Here example is BRCA1, TP53, and HER2. So maybe we can proceed with this web tool, and here you will see once it's loaded. So this page is loaded, and maybe we can try out something. So find me genes associated with uh, liver cancer. So it will basically take a few moments to explore the web and different papers, and it will give us the update. So it will take a few moments, and it will give us the output in the form of summary. So you might have to wait for a while, and here it is. So it has searched 10 different literatures. You can also extend your uh, required amount of um, publications that you want to explore, and here it will give a summary of the different papers what is associated with it. Then you can do you can check out different filters like publication and the Q1 Q2 journal. So it will filter everything which the option which you don't get in other LLM or AI tools. So this is Elicit. Now we can discuss about the next uh, tool. So which one is PubDater? So PubDater is a biomedical text mining tool. Similarly, we can use it for literature reviews. And so basically it automatically identifies the genes, diseases, proteins, mutations, and how we can use is we can either paste the PMID or gene name or the disease name, and it will give us the results. So if we go to this web tool, so you can see there are certain examples. So like maybe we can search about olaparib, which is one of the most common drug used in cancers. So here you can see uh, this has a similar interface like as Google Scholar, but you can definitely explore out like what exactly you are looking for. So it's basically offered by National Institute of Health and National Library of Medicine, which is basically from NCBI of USA. And you can of course try it out and explore what are the different tools here. So next one we'd like to visit is the other web tool, which is called Cite. So it's categorized as citation intelligence, and also it is a similarly a literature survey tool. So basically you can check whether a paper supports the contradictions and mentions and other publications. So basically you can check multiple different things. So if we go back to this web tool, so you can see uh, like we can, let's say we'll search one of the example given here. So how do TP53 mutations affect the immune checkpoint inhibitor response in metastatic tumors? So basically it will take a few time, and it will do the search operation across the web, and here you can see what are the different citations that have been already been used, and which you can explore out around. So maybe you can explore this thing, and maybe you have to get a premium version for this to access more of the contents. And so we'll skip it from now, and we'll move to the other one, which is basically ensemble. So ensemble is this is how the interface usually looks like. So it provides the gene sequences, protein sequences, variants, and gene annotations. So you can access the updated genome databases and download the FASTA, GTFs, and all. So basically, it's an updated version of the original ensemble database along with certain other tools. So let's explore this web tool. So here you can search the human, and you can search TP53 or any other gene, and hit enter. So it will redirect to the another page, but currently there is some errors, which is why this page is not loading. But overall you can see this should load some uh, information related to this gene and the organism. So maybe we can cover this tutorial in another video. The next one which we have is the biopsych, where you can you can use for metabolic pathways, enzymes, and metabolites. So if we click on this web tool, and we'll get an interface like this, and we can put some uh, metabolic pathways. Here I am putting this AMPK. It's also a protein, and it also has a role in AMPK pathway, and we'll see what we can uh, retrieve from here. So basically it gives the search results for AMPK using this database, and we have certain matches related to this, which is basically PRKAB1, and we'll see what is this. So here AMP activated protein kinase B1 is there. So it's basically a protein complex, and here you can see the different other informations in related to this. And you can of course use this all these features in this web tool, and we'll definitely move to the next one as of now. So this one is alpha missense, which is basically by Google DeepMind. So here you can put a reference DNA protein, and it will detect the missense protein, and it will give you in the output in the form of this. And we can click on this, and it will give us this interface. We can try it out in some other web tutorial, because it will be of course long video. We have this deep variant, which is basically developed by Google. So it's available in GitHub, and we have to use this in our local machine, and we can use this for alignment. We can use this for variant calling, and the input will be alignment, BAM, or CRAM files, or the FASTA files, and we can definitely follow the workflow to get these kinds of results which we are looking for. The third one, the next one is the Splice AI, which is basically for splicing prediction. So we can use this web tool, and see the different uh, diseases and mutation, and the splice variants, like whether the mutation affects the RNA splicing or not. And if we proceed to this web tool, so here you can enter this different examples, like you can give them show more examples, and suppose we can copy this one and click submit. So it will give us some details after a couple of minutes, a couple of seconds, and from here you can basically use this information to interpret the different results. So these things we can cover up in some other videos. The next is the RNA composer. So this tool is used to predict the RNA structure. So here example, you can see we have this 3D RNA structure, which basically you can of course download. This is for visualization and prediction of other structures and their interaction. The next one is the DeepLog2, which is basically protein localization web tool to predict the eukaryotic protein subcellular localization, and uses the deep learning model. So here you can see this is the interface of this web tool. So this is the link of this web tool, and here for example you need to enter the protein sequence in the FASTA format, or definitely you can choose file, or you can load an example and you can submit, and it will take a few seconds or maybe few minutes, and it will give us the output. For now we will be skipping this and we'll proceed to the next one, so which is basically the AlphaFold. This is one of the popular web tools which basically uses AI and machine learning algorithms to predict the protein 3D structure, and it has very high accuracy. So it is also basically developed by this Google. So Google DeepMind is basically one of the biological resources which basically develop this uh, web tool. And if we go to this web tool, we can see this is the protein structure database, and developed by Google DeepMind along with the EMBL and EBI. So here you can simply click on the examples, and it will take a few moments to give the results, and you can click on them. Again, here it will take some moments to generate the structure and predict the different parameters associated with this. Uh, Now we'll move to the next one, which is basically Robeta. So Robeta is a protein structure prediction tool, and it's used for alternative protein structure prediction, and it has advantages like comparative modeling and de novo prediction. So this is basically the web interface of Robeta, and if we go to this web tool, so you can see that results will be available to download until this, and so basically uh, here in the structure prediction, you can check the example results. These are all the examples for your personal work. You need to register and log in, and then you need to proceed with the pipeline. So here you can see the amino acid sequence which has been displayed, and the other parameters. So this is basically an example, and you can see what are the different things that it's loading. So it's taking some time to load, but we already have the screenshot here, so it will show a protein like this. So the next one is we have protein mpnn. So we can use this tool for designing different new proteins, engineering and optimization. Uh, so in this tool, basically, it claims that uh, new protein structures and uses neural network to create new protein structures. So similarly, like designing the different new protein structures, this tool can be used. We have string, so basically we have designed different proteins and protein structures, but what is the different interaction of this protein structures? So we can use this web tool called string, and it's a very wonderful web tool where you can use this. For example, I would check TP53, and organism maybe will keep Homo sapiens. Anyways, it will definitely do the auto detection, but so let's see if we can proceed with this. Here you have to click continue, and it will show the different proteins which are basically connected to this protein called TP53. So here you can see that TP53 is having maximum interaction, and that's why it is a very densely connected network, while there are other uh, uh, proteins which are connected to this. So you can do different analysis, and you can export this to your cytoscape tool, and do the clustering analysis, and you can visualize them in other different uh, networks or experiments or other different forms. So I have already uh, discussed about this tool in my previous YouTube tutorials. Maybe we can design some new other tutorial where we'll discuss about using this all these online tools. As of now, we'll move to the next one, which is BioBart PT, and it's available on GitHub, and we have to use this. We have to download this, and we can use this for. It's basically a language model, biomedical language model, so it can extract the biomedical information using the AI. So we have Path AI, which is an online tool which is basically for digital pathology analysis, and it uses the AI for analysis of the different pathology sites. We have Deep Cell, so it's an interesting tool. So if we go to this web tool, and we can click on data or predict. So if you have some images, you can simply click on predict. You can upload those microscopic images and it will predict the image types. So for online data, you can see, you can download this data, you can use them for further analysis. For example, you can download a data set and use this for um, like AI ML prediction project, and this is quite useful tool. So moving to the next is Metabo Analyst, where we can work on the metabolics and metabolomics. So this is basically a statistical analysis tool, and it has heat maps, principal component analysis, and pathway analysis. So we can click on this, and you can check this web tutorial. So they have different tutorials for different analysis, so you can definitely see and use those tutorials for your particular project. And there you can see different analytical tools that they have displayed, and you can always use them for your personal research work. Moving to the next, we have MOFA Plus, or MOFA, which is basically multi-omics factor analysis. So it is basically used for multi-omics integration and integrates the RNA, proteins, and metabolomics. It also offers the heat maps, and it's advantageous for biomarker discovery and data integration. Then we have XPACY, which is basically from SIB, and it's a protein bioinformatics resource. It's used for protein analysis, and you have this molecular weight and PI and other domains and other parameters which you can check in this. So this itself is a long tutorial, and we'll definitely cover this in some other tutorial, or you can follow other online resources. We have SIB, or Swiss Bioinformatics Resource, where which is basically the SIB SWISS. So basically it's from Swiss Bioinformatics Institute of Bioinformatics, and it's used for collection of the bioinformatics tools, and advantages are like protein genomics and other databases are there. And then we have this ESM metagenomics. So it's again a protein structure prediction database, so a protein structure database, and it is used for protein structure prediction of from metagenomes. We have this CRISPR net for genome editing, and it's available on GitHub. We have to download this package, and it's available for both Python and R, and we can use this sorry, it's available only for Python, and we can use this for designing the CRISPR and guide RNAs for different genes. We have this tool called DeepCam, which is basically AI aided drug discovery tool, and if you click on this, you land on this web tool. So you can check this out, and we have these tutorials for how to use this DeepCam. So basically you can predict the chemistry structures or chemical models of drugs or other small molecules. So I think this we can use on both R and Python, or for sure we can use this for Python, because it's again a machine learning model, and you can try this out. And we can check it in other subsequent videos. So another drug discovery tool is Atomwise, which is right now it's called Numerion Labs, and you can visit their web tool, and they give the they have the advantages like drug repurposing, virtual screening, and for example screening against EGFR. It's basically AI based molecular docking tool. Now we have Neurosnap, which is categorized into neuroscience, and it has an AI assisted neuroscience data analysis and visualization with utilities that support the general bioinformatics workflows. So it simplifies the complex analysis, interactive visualization, and it's easy for beginners. We have this Eterna, which is basically RNA drug designing game. So you can click on this thing and you can solve puzzles and these things. So basically it allows you to solve puzzles to advance the research using RNAs. And of course you can connect to the community and learn, and you can play around and design different RNA, and as they say, address research problems. So you can check out the publications and technology they are using, and explore all other resources which are there in this web tool. The last one, not last, it's a prompt bio, and we have one more, we have two more. So this is prompt bio AI. So it's again an AI tool which generates the prompts effectively, and improves the AI assisted analysis, and helps in literature review, coding, and data interpretation. And you can check out this web tool, and you can see what are the free resources that you can use for your project, and what else you require. You can maybe you can purchase or take subscriptions for your analysis and all. The last but not the least, it's which is prompt bio. Sorry, it's OMA, or the orthologous matrix, which is a project which is used as a method and database for interference of orthologs among the complete genomes. uh, the purpose of this is that it can run on custom genomic transcriptomics data, and it uses the OMA standalone software, and is possible to combine pre-computed data with custom data by exporting the parts of this OMA database. So you can click on this link and you can see this database allows you to work on different species and the different genes, genomes, proteins, and multiple other things. Again, it's from SIB, or Swiss Institute of Bioinformatics. So basically, it will have multiple data from this database, and it also uses the AI and machine learning to interpret multiple genomics and transcriptomics data sets. So this was all about our different web-based AI tools. So in the upcoming videos, we'll definitely check out one small project which we can use, in which we can use this online web-based tools for different analysis. So till then, stay tuned, and thanks for watching this video.